Good evening, everyone. Welcome to NeuroFutures 2016. Please take your seats. The program's going to begin momentarily.
Wow, everybody went quiet. Okay, can everybody, oh, you can hear me. I can even hear myself. So welcome everybody to the kickoff of the 2016 NeuroFutures Conference. We're extremely happy that we're able to host it here at our new Allen Institute building. And hopefully over the next couple of days, those of you who are going to be hanging out with us will get to see a little bit more of what the place looks like and what we get up to here when we're not pulling you out of a sunny Sunday afternoon in Seattle. I'd like to, up front, even though we'll do this in the morning, thank the different organizations that have helped pull this together. That's Oregon Health Sciences University, University of Washington. For the first time, UBC, University of British Columbia, were more involved. And of course, the Allen Institute and various branches of the Northwest Neuro Neighborhood that are providing our moderators and numerous helpers you'll see around. So tonight's lecture and panel discussion is going to kick off 2 days of what we hope are going to be a pretty exciting and interactive time for everybody here. So in getting ready to introduce Anne Churchland, I was thinking this morning, what do I say? I'm like, okay, it's Father's Day. So thank you to all the fathers in the audience who made the decision to come here tonight. Some of you have brought your children, and I thank you for making that decision too. And when I thought about that, I thought everybody who's here tonight is here because of a series of decisions that have been made, either by themselves or by other people. The decision your parents made to have you. The decision of your parents to choose where you would grow up and which school you would go to. Your decision of what classes to take during the course. Your decision to listen to teachers and profs that inspired you to follow a particular path that would take you down a career path that you decide. And then maybe change based on your own decision. So it suddenly struck me that decisions kind of rule our lives. The decision to make friends with somebody, fall in love. The decision of who to vote for, which I think is a little easier this year than most years that we've had to deal with in a presidential election. The decision to buy an assault rifle and the decision to use it. These are all decisions. Decision-making is a process that governs so much of our lives, and yet we know so little about how it happens. But that's changing, and it's changing in large part because of the superb work being done by our keynote speaker tonight and her colleagues in her field. So Anne Churchland, Dr. Anne Churchland, who's about to speak, is a leading light in the neuroscience of decision-making, which is something we all have, I think, should be enthusiastic about learning more about. She's also, and I hope she won't mind me mentioning this, a member of a neuroscience dynasty of a sort. I have no doubt that some of her own decisions were influenced by growing up in a household where her mother literally wrote the book on the computational brain about 30 years ago, maybe more, and is a leading light herself in the field of philosophy of mind. Anne herself has chosen a different path. But many of us, I think, when our children hear our conversations, get pulled in to wondering about how they follow their own questions and their own curiosity when they're put in that kind of environment. So what Anne has done is she's worked her way through different areas of neuroscience, working with Stephen Lisberger at UCSF, learning about the regulation of the visual system, moving up here to UW to work in a non-human primate model at UW with Mike Shadlin when he was here, and then moved across country to Cold Spring Harbor Labs to set up her own labs. For those of you not familiar with Cold Spring Harbor, it's also began as a nonprofit, philanthropy-driven research institute that has produced many prizes, some of which have been awarded to Anne in her early career. So in her early career, her research became recognized and awarded with a long series of names that we revere: the McKnight, Merck, Klingenstein-Simons, and Pew Foundation. These are all awards that are given to early career investigators that people are expecting to shine. And that is indeed what has happened and what we expect to keep happening. So from here on out, I'm going to hand the floor to Dr. Anne Churchland, and she can fill you all in a little bit more about how you might make some of those decisions that you were never sure about, how they were coming about. And please, I know some of you might be watching NBA Game 7 in the background. Put your phones on silent. Thank you. Anne.
Okay, thank you very much for that introduction, Jane. And thank you to the Allen Institute and the other institutes and foundations for organizing this meeting, and also for inviting me to come and speak. So I am going to tell you today about decision making, and specifically how brains combine multiple pieces of information to guide decision making. So just stepping back from decision making for a moment and thinking about the field more broadly, the mysterious inner workings of the mind have really captured people's imaginations since the beginning of time. And this image here shows Empedocles the Greek. And during his time, he didn't have access to the same kinds of tools that we have today for measuring and manipulating neurons to understand how they work. And instead, he had to rely on observation and introspection. So for example, he had this idea about how vision worked. He thought that maybe there were beams of light that were shone out from the eyes that hit objects of interest that a subject might be looking at. And of course, we now know that actually it's exactly the opposite of that happens. There's light that comes from the objects and into the eyes and hits the photoreceptors and so on. But back in Empedocles' time, that was really the best tool that was available, that is, thinking about it, for figuring out how the brain and the sensory systems might work. And there were other kinds of people that could gain some insight into how brains worked. People knew, for example, that patients might suffer from epilepsy that would temporarily make them unconscious and also lose motor control. And that afforded some insight into what brains might be doing for our bodies and ourselves, but was really very limited. There were some additional insights that were provided about how brains might work from mind-altering substances like poppies and later on alcohol. And by observing that certain kinds of substances can alter brain function, that also gave some insight into how brains might work, or how they might work when they're in an altered or different state of some kind. And indeed, this has interested people interested is maybe not the right word, for a long time. People have been using alcohol since about 5000 BC. That's the estimated time when people began to use alcohol. And who knows, perhaps it went back even further in time than that. But we now live in a really different era where we don't have to use just observation and introspection to learn about the brain, and we instead have a series of pretty powerful tools that will allow us to gain far more insight than was previously possible in any of these preceding centuries. And in the last kind of 100 years or so, these technologies have been developing at a really, really rapid rate. So this is an image from David Hubel and Torsten Wiesel, who got the Nobel Prize in Physiology and Medicine for their discoveries about how neurons in primary visual cortex work. And you can see an image of a cat up there because they mostly studied neurons in a cat brain to understand what happened. And they used a technique that's a classic technique, but still very popular in the field today, called electrophysiology. And that simply takes advantage of the fact that neurons communicate with each other using electrical signals. And so by putting even a very simple fine wire into the brain, it's possible to measure those electrical signals, and in this case, gain insight into what the neurons are doing. So they learned a lot about those neurons by showing different kinds of visual stimuli to the cats and looking at how the neurons responded. And this was a big step forward, using this technique to see what the neurons do to measure their responses under different conditions. This is another really transformative technique that probably you're all familiar with. This is a functional magnetic resonance imaging, or fMRI, in which human subjects are put in a large magnet that makes it possible for scientists to visualize or observe their neural activity or at least regional cerebral blood flow, which is taken as a proxy for neural activity, as human subjects are doing different things. So scientists can compare, for example, the blood flow during a resting state with the blood flow during experience of a visual image, with blood flow during a decision, and gain insight into what kinds of parts of the brain might be involved. And this, again, has been really transformative in a sense in that it's allowed us to look inside human brains and see what's happening during normal brain function, and also how activity might change during disrupted brain function, disease states, and things along those lines. It's also a coarse technique in a sense because the smallest unit of resolution, that is a pixel, is thousands or sometimes even tens of thousands of neurons. And so if you have a part of the brain where neurons are heterogeneous, that is, all the neurons are doing different things, then if you see the average signal of many thousands or tens of thousands of them, it can be difficult to get a clear picture of what's going on. In this slide, you see another imaging method, but this is at a much finer spatial scale and also finer temporal scale. And this is called two-photon imaging. It's a technique that we use in my lab and also is used extensively here at the Allen Institute for Brain Sciences. This particular image is taken from a group at Janelia Farm, and each one of these little circles or dots that you see is an individual neuron that's expressing an indicator that allows this group to measure the neural activity that's taking place in the brain of this animal. And in this case, this is a larval zebrafish, so it's a very little tiny fish. But what's really neat about this setup is that the scientists are able to measure the activity of every single neuron inside this animal's brain as it's doing different things. And that capability to measure the entire brain, every neuron in the brain, while animals are doing different things, really catapults us to a completely different place compared to people in the many centuries leading up to the present time, when the tools for understanding the brain and understanding the mind were very limited or indeed non-existent. So these tools are really helpful, and they allow scientists to achieve many different goals. And one main goal is to understand the neural circuits that give rise to behavior, to connect what's happening in the brain to what's happening at the level of behavior. So why is this important? Well, I think really for two reasons. And one of them is that one could argue that this is really one of the big basic science problems of our time. This is an incredibly interesting and important intellectual question. What is it about our brains that makes it possible for us to do the things that we do? And it's also really important for mental health reasons as well. Brains can be disrupted in any of a number of ways. This is a painting that is from Vincent van Gogh showing a man who's suffering from depression. And depression and mental illness in general are really a huge burden for society. It's estimated that 1 in 17 people suffer from a major mental illness, and that 1 in 19 suffer from addiction. And this is a huge cost in terms of providing care and services for patients. And I think, moreover, there's an enormous personal cost, as anyone knows who's either suffered from mental illness themselves or has been around a loved one who's struggling with these issues. So it's for both these reasons that I think it's really critical to take advantage of these tools and to use them to unlock the mysteries that people have been wondering about really since the beginning of time. So let's kind of take stock of where things are in the field. And I won't have time to do this very extensively, but just to give you kind of a sense of the upshot. Well, how are things going, right? So now we have all these tools. So what have we figured out, and what's still remaining? Well, so when it comes to certain kinds of problems about linking brain and behavior, we're actually doing quite well. Okay, so when it comes to understanding reflexes, we've pretty much nailed it. So I started with this one because it's a really simple reflex that you probably all know about. It's called the knee-jerk reflex. And when your doctor hits the knee with the hammer, your leg reflexively will kick up. And we know all of the neurons that are involved. We know it just goes through the spinal cord. We know pretty much everything about it. It's completely well understood using, and has become well understood using some of the techniques I've mentioned throughout the talk, electrophysiology, as well as a few other classic techniques. Here's an example of a second reflex that's super important, but actually most people haven't heard of it. It's called the vestibulo-ocular reflex, or the VOR. And the point of this reflex is to keep the world stable on your eyes as your head and body are moving through the world. So just to prove to you guys that you all have one, we're going to do an audience participation demo. So what everyone should do is hold up their finger about 6 inches in front of their eyes and look at your finger, and then move your head back and forth about the rate that I'm doing right now. And most people will find that overall, as they look at their finger, that it's fairly stable on the retina. It doesn't look super blurry. So now for comparison, take your finger, hold it again about 6 inches away, and this time keep your head still and move your finger back and forth. And try your very best to follow it with your eyes as fast as you possibly can. And most people observe that even when they really try their best to follow it with their eyes, that the image looks very, very blurry on the retina. And that's because your eyes are not very quick. The ability of our visual system to track moving objects is quite limited. That's for a variety of reasons, including the fact that the eyes themselves are just really slow. Light comes into the eyes and gets kind of stuck on the retina for tens of milliseconds for reasons that I can talk about later. But fortunately, this reflex takes advantage of the fact that the sensory systems in your inner ear are super, super fast. So remember the first time when your finger was still and you moved your head back and forth, when you move your head to the right, the fluid in your inner ear detects that motion and sends your eyes in the opposite direction with a 12 millisecond latency so that your eyes move just the right amount to counteract the head movement and your finger stays stable on your retina. So this is a super important reflex for keeping the world stable. And it's also one that's extremely well understood. We know all of the neurons that are involved. It's a pretty simple three synapse circuit. We know every piece of it. And it's so well understood that, in fact, people are beginning to use the VOR to understand how neural circuits can change with experience. You can imagine, for example, if you're wearing glasses that make the world bigger, you'll need to recalibrate your vestibular ocular reflex. And humans and animals are pretty good at doing that on the fly. And neuroscientists are taking advantage of this extremely well understood system to evaluate how it changes. So again, this is an example of a reflex, and we pretty much nailed it. It's pretty much done. We understand how it works really, really well. But I would contrast that with other kinds of behavior. And just to schematize this is a schematic of a rodent. This is a brain from the Allen Institute Common Coordinate Frame. And we can look inside the brain of these animals using the 2-photon imaging that I told you about a moment ago. And this is a movie that was taken from my lab from a mouse that is passively looking at visual stimuli. And you can see that there's lots of neural activity. There's about almost 1,000 neurons in this visual field. You can see that they're flickering on and off on some kind of time scale. And we measure this neural activity. We know something about what the animal's doing. We know about the visual stimuli that we're presenting to the animal. But even though we have these measurements, there's so much that we still don't understand. So we don't understand how these neurons encode information. We don't understand how they make decisions. We don't understand how they represent time. And we don't understand how the neurons in different parts of the brain interact with each other. And those are just a few examples of really big problems in neuroscience that we're beginning to make traction on, but that we still are a long way from fully understanding. And if our goal, which I set out at the beginning, is to understand how the brain drives behavior, well, these are really fundamental pieces of that understanding. If you want to understand how the brain supports behavior, you need to know how it represents time, how it encodes information, how it integrates information across different kinds of brain systems, and so on. So you might wonder, well, why exactly is it that these questions are outstanding, right? Because I've argued that, well, we have access to these incredible technologies that make it possible to measure lots of neurons. So what's going on? Why are these questions, encoding information and time and so on, why are these such difficult problems? And there's a lot of reasons. So I'm going to tell you what 4 of them are. And the first one is that brains are complicated and have a lot of neurons. So this is one section from a mouse brain. It's about 50 microns thick, which is about as thick as maybe one of your hairs. And this is stained with a particular substance that makes the neurons purple. So every time you see a purple dot, that means that there's a neuron there. And as I zoom in, you can see that there's kind of an interesting structure to the brain. And I'll zoom in even more. And you can see that even in this one very, very thin slice of one part of the brain, that there's lots and lots of neurons, right? There's a lot there. And this is from a mouse brain. They have about 100 million neurons in their brain. So that's a lot of neurons to understand. Maybe we don't need to understand all of them at the same time. Maybe we do, nobody knows. But that's a lot of neurons. And that's in a mouse brain. So in a human brain, we have about 86 billion neurons. And that just highlights that these are really, really complex organs. And it isn't just the number of neurons that makes them so complex. So in addition to having lots and lots of neurons, I'm going to put next to this section a section from the same mouse, but from another part of the brain. So the one on the left, that's from a section that's about here. You can see the very anterior part of the visual cortex up here, and this is the hippocampus. And this section here, this is from further back, a part of the brain called the cerebellum. And it's really obvious just by looking at these two sections that these structures are really different. And so even if you were lucky enough to figure out what's happening in these kinds of circuits in this part of the brain, they might be fundamentally different from what's happening in the cerebellum. There are different kinds of neurons. They connect to each other in different ways. They're connected to different parts of the brain. And so because there are so many different parts of the brain, understanding how they work together is quite challenging. So that's the first problem. Okay, and the second one is that in thinking about how to go about solving the mysteries of the brain, it's tempting to kind of rely on introspection to try and figure out where a good entry point is. And the trouble with that is that often our introspections about brain function can be extremely misleading. So here's an example of that. So when people first started thinking about what it would mean to understand the brain, they sort of thought, well, you know, when we really have achieved a high understanding, we'll be able to understand the truly kind of the pinnacle of cognitive function. And that's got to be something that only humans could do, and maybe even something that only the really smart humans could do. You know, one of the pinnacle of cognitive function, it's got to be something like chess, right? Like chess is hard. You have to work really hard to get at it. You know, that's something, if we could understand that, we'd really be a long way to understanding how the brain worked. And people kind of had these sorts of intuitions back in about the 60s, and it seemed that, for example, teaching computers to play chess would be almost an insurmountable problem because it's such a complex cognitive task. But that just turned out to be wrong. So computers, the fast computer Deep Blue beat Kasparov many years ago at this point. And just recently, Google DeepMind had computers that beat human players at Go. And so there's been the ability of computers to beat humans at playing what we consider to be incredibly difficult games. Well, those were hard problems. It took a lot of work to solve them, but they were pretty tractable. Like, we pretty much nailed that. Computers are better than humans now at even those games that we hold dear as being kind of the pinnacle of human cognitive existence. But by contrast, so on that one, kind of the computer is winning. But by contrast, the ability to just pick up the chess pieces and move them from one place on the board to the other, the computers are doing terrible. And any 6-year-old is much better at even the very best robot at just picking up the pieces and moving them from one square to the other. And that's because the problem of motor control, the problem of designing a robot, or even understanding how a human can take a complex piece of machinery like an arm and move it to the right place and put the hand around the piece and pick it up, that turned out to be a really, really hard problem, much harder than anyone anticipated when they first started thinking about what it would mean to understand the brain. And so just to make that perfectly clear, you're going to see here a movie of a state-of-the-art robot from the DLR Institute of Robots and Mechatronics. And this robot has learned to pick up this cup. So it gets its fingers in the right place. Closes the hand, and then slowly picks up the cup. So, like, this is not bad, right? This is hard. It's hard to get, just the right pressure so you don't break the cup, but enough pressure so you don't drop the cup. And you see this, and it's kind of tempting to think, like, wow, robots, yeah, pretty good, right? But just for contrast, this is the World Cup Stacking Championship. And she won. She got the world record. So I think these two movies, seeing them side by side, illustrates how far we have to come in our understanding of an ability that all of us take for granted all the time, the ability to move our bodies around and use them to interact with the world. It's hard to build robots that do that, and it's really hard to understand how that happens in human brains. So that's the second reason why I think it's been a really challenging problem, that our intuitions can be misleading. A third reason is that efforts have been somewhat uncoordinated. So there are neuroscientists now all over the world in almost every country, and many of them are kind of working as separate independent labs. And it's very difficult to coordinate efforts across different groups there could be two groups, both of whom are using a pretty similar technique and asking a related scientific question, but because of small differences in the way they collect their data and the way they analyze their data, it's very hard to make sense of their findings. And you might have two groups that just have completely opposing findings. And as an outsider, maybe reading their papers or hearing their talks, it's very difficult to understand why. And I think that's starting to change. There are groups like the Allen Institute where there's a lot of people working collectively on a single problem. And there are other consortiums that are starting to develop within neuroscience where larger groups of labs will come together and try to coordinate their efforts and standardize their techniques, and also to share their data so that it's possible to have a more coordinated effort. But that's something that's only just now beginning to happen. And for a long time, the field of neuroscience has been a lot of kind of independent labs working on their own. And that's probably not the most efficient way to solve the problems that are of this magnitude. And the final reason is that the right theoretical frameworks for understanding the brain are still being developed. So it's true that at this point in neuroscience, we can collect large amounts of data. We can measure the responses of many neurons at the same time, certainly in the level of thousands. For other kinds of setups, maybe even at the level of tens of thousands. And we can monitor their activity as it unfolds over time. But that is not enough. In order to understand what's really happening in the brain, it's essential to have the right theoretical framework to understand how those neurons are working together, how their activity as a population is unfolding over time, how they connect to other parts of the brain, and so on. And this image kind of schematizes a data pipeline, which is from a paper that I wrote recently as an introduction to an issue on neural computation, kind of highlighting what happens with data. So we start with raw data. We process it to extract time-varying estimates of individual neurons. We reduce the dimensionality using different kinds of approaches, maybe averaging or something more sophisticated, principal components analysis, machine learning. And then eventually connect that data. We link it to models to establish what the mechanism is. And theoretical neuroscience is a rich field and has a history of having a strong presence in the field of neuroscience. But at this moment, we need theoretical neuroscience more than ever. And we need theorists and experimentalists to come together to work as a team to understand the large data sets that are unfolding. We need to move beyond the level that we're at now, which is where sometimes these enormous and incredibly rich data sets get reduced to kind of pie charts. You know, this percentage of neurons did this percentage of neurons did that. That's really standard in the field, and that isn't really enough to tell us how the brain is working and to make it possible to achieve the goal that I laid out, which is to connect the brain and behavior and understand the neural circuits that make that happen. Okay, so I started out the talk by telling you that I think there's a big gap in our understanding of sensory motor reflexes, which by and large, many of them we understand very well, and our understanding of truly complex behaviors, like how do neurons encode information, how do organisms make decisions. And I've told you that some of those reasons are that brains are complicated, that introspections can be misleading, that efforts to understand the brain have been somewhat uncoordinated, and finally, that the right theoretical frameworks are not yet fully developed. So in developing my own research program, my goal was to bridge this gap by studying decision making. And in thinking about what was the right way to do that, to bridge that gap, I thought, well, what really distinguishes the kinds of complex behaviors that we might want to understand, those kind of big life decisions, some of which Jane talked about in her introduction? How do we bridge the gap between those kind of complex behaviors and simple sensory motor reflexes? And I thought, well, what are some of the features that distinguish complex behavior from the simple reflexes? And a few things, I think, to me, come to mind immediately. And the first one is flexibility, especially in time. Okay, so when you're at the doctor and they hit your knee with the hammer, you don't have any control over when your leg will make that response. It happens immediately with a very short latency. And similarly with the vestibulo-ocular reflex, when you move your head to the right, your eyes move to the left with a 12 millisecond latency, and you don't have any control over that whatsoever. You couldn't suppress it even if you wanted to. Even if you were in the dark, your eyes would go to the left with a 12 millisecond latency. And there's a good reason for that. Those reflexes are designed to do very specific things, to keep the organism alive and allow it to be able to see things in the visual field and so on. But with complex behavior, things operate on a much more flexible time scale. So for example, we'll take kind of a quintessential real-world decision. You're deciding which car to buy. Well, it's not like the reflexes where there's a particular moment where action kicks in. You can make that decision at any moment, right? You might decide it after seeing one car. You might spend all day looking at cars and decide 5 months later which car to buy. You have all kinds of information coming in, and you decide flexibly how you want to use it. So that flexibility in time is, I think, the first thing that starts to distinguish complex behavior from sensory motor reflexes. And a second thing, I think, is that often in truly complex behaviors, we integrate information from lots of different systems. So again, with the car analogy, if you're deciding what kind of car to buy, you're going to partially be informed, for example, by information coming from memory, maybe what you've read about the car or heard about the car. You'll certainly be informed by your sensory system, what the car looks like, what the car smells like. I think Linda Buck in her talk tomorrow will tell us how the brain figures out how to use that olfactory information to judge many different qualities about the car, and certainly its smell. It can be quite informative. And all these different pieces of information, the memory, the vision, the smell, they all are put together. Oh, go back. They all are put together to allow us to make good decisions about what to do. So it's really those two features, the flexibility in time and the ability to integrate information across multiple senses, that is what starts to distinguish sensory motor reflexes and complex behavior. So in thinking about what sorts of behaviors, I wanted to have behaviors that combine some of those features. And when I was starting my lab, I thought, OK, this is what I want to study. So I'm going to go to the human literature. And I'm going to look for kind of interesting human behaviors that are really well studied, and hopefully ones that have a useful theoretical framework, because that can be a helpful tool as an experimentalist for interpreting the data and so on, and see whether I can't find some kind of interesting behaviors that capture some of these features, and then bring those to a tractable experimental model, in my case, rodents, which is what we study in my lab. So one of the behaviors that I found immediately interesting was multisensory integration. So integrating multisensory information, and mostly I'll talk today about visual and auditory, although there are lots of other kinds of multisensory integration. It's something that we do as humans very, very naturally. We do it all the time, often without thinking about it. And that's because most stimuli in the world are just inherently multisensory. When you're talking to somebody, we tend to think about the auditory signal as being the most relevant one if they're talking, for instance. But you get tons of information from your visual system as well. If there's a lot of background noise, you get a much clearer picture of what they're saying by looking at their lips. And people's gestures and so on provide additional information as well. And that's true of many kinds of stimuli in the world. It's a very natural thing to merge information across sensory systems. So this is an example of a multisensory illusion, and there's a few of these. I'll give you a couple of examples. It's one that you're probably all familiar with. This is a ventriloquist. And so even though the sound is coming from the speaker's mouth, it really appears to emanate from the mouth of the puppet that's somewhere between 6 and 18 inches away. And part of the reason that this happens is that as primates, our spatial vision is really good. Our spatial acuity is extremely high. So we're really good at telling where things are in space with our visual system. And so because the puppet's mouth is moving in a particular way, our visual system tends to dominate in that particular setup. And I should say there's actually lots of situations like this one in which vision wins. In fact, you're all experiencing kind of an extreme ventriloquism at this moment, in the sense that your visual system is probably looking at me, but the auditory cue is coming from the speakers that are over there. But I'm guessing nobody thought about it. Like, probably nobody, when I came up here and started talking, was like, wow, this is crazy. Like, her voice is here, but the sound is there. It's blowing my mind. Because your mind just naturally goes with the visual cue, because our spatial vision is so good. And we're not that bothered when auditory cues don't come from exactly where they're supposed to come from. So this has been known for quite a long time, obviously, because ventriloquists are good showmen and are very appealing. And people thought for a long time that the reason that their trick worked, that ventriloquism is so compelling, is because as humans we're what you'd call visually dominant. Certainly we have good visual systems. Maybe it's just the case that whenever audition and vision are in conflict, we just go with vision, because we're good at that. But that turned out to not be right. So now I'm going to show you another kind of example of a multisensory illusion. And now you guys are going to be subjects in what we call a psychophysical experiment. So we're going to measure your perception. So your job here, there's going to be a flash that appears on the left side of the screen. It's going to be a black dot that flashes on and off. And your job is to say whether you see 2 flashes or 1 flash. OK? So raise your hand if you saw 2. OK, most people saw 2. OK, so now say how many you see for the next one. Raise your hand if you saw 1. OK, yeah, so this is pretty typical. Most people see 2 on the left and 1 on the right. In fact, those 2 stimuli were identical. And in both cases, there was only 1 flash. So I'm going to show you again without the sound. So that was the stimuli that you all judged to be 2 flashes. There was only 1 flash. And this one is the one that you correctly judged as 1. There's only 1 flash. And the difference between the 2 is the one on the left was accompanied by 2 beeps. So there was 1 flash, but 2 beeps. And in that case, your auditory system dominates. So I'm going to play with the audio on one more time. It's going to be on the left. And one of the things I find compelling is I've looked at this thousands of times, and it's still exciting too. I can't suppress it. So this is really the opposite of the ventriloquist, right? So remember, with the ventriloquist illusion, it's cue conflict. So audition and vision are telling you different things, and most subjects go with vision. This one is, again, cue conflict because you have 2 beeps and 1 flash. But this time, almost everybody goes with audition, right? Most people report 2 flashes, even though there's only 1 flash. And this kind of blew people's mind when LaDawn Shams, who's at Berkeley, first developed this illusion, because people really think of humans as being visual. But what she argued was, no, that's not the right way to think about it. It doesn't make sense to think of organisms as being visual or auditory or whatever. It really depends on the nature of the decision at hand. So in this case, you're relying on a judgment that has to do with time. You're not saying where flashes occurred, but when they occurred. And the auditory system is really, really precise at telling you when things occurred. The cochlea is super, super fast. But as you'll recall from the demonstration with the VOR, your visual system is really slow. So when you have to make a judgment that has to do with when things happen in time, you rely much more on your auditory system because your visual system is just not nearly as temporally precise as audition. So these are 2 examples of extreme cue conflict situations. And some people were inspired by this and wondered, well, maybe we go kind of smoothly from being completely dominated by one sense to being completely dominated by the other sense. We make that transition continuously, depending on how good each modality is at the current moment. And so this is an image. It's kind of a strange image, but it's from a really landmark paper from another group at Berkeley, Ernst and Banks, that was inspired by work by Mike Landy and others. And in this one, they had subjects making judgments about the height of a raised bar. So they had a visual cue and also a haptic one. That means the subjects could feel it. So there was a bar there, and the subjects could feel it, and they were supposed to say how high it was compared to a memorized standard. And then the visual cue they got was a random dot stereogram. And for those of you that don't know what that is, there are 2 little images of dots, one shown to each eye, and subjects kind of fuse them the way that you do with a 3D movie. And that gave subjects a visual estimate of the raised bar. And here's the key thing that these guys did. They could independently vary the reliability of the visual cue. So sometimes they did a really good job with the 3D image so that it was very clear the height of the raised bar. And sometimes they added a bunch of noise dots that didn't really fit with the image at all, so it was much harder to make a judgment about vision. And what they found is that subjects smoothly transitioned from relying exclusively on the visual image when the visual cue was reliable, to relying on them equally when the vision was a little less reliable, to relying completely on the haptic cue when the visual system was not reliable at all. And this suggested that the way people used to think about multisensory integration was totally wrong. It's not kind of an all or none thing where you go with the vision if you're human and go with something else if you're not. Instead, it appears that our brains are able to estimate, probably on a very rapid time scale, the reliability of individual sensory cues and use that to inform the way that they're combined so that information is never thrown out. So that if the visual information is, not amazing, you still use it, you don't throw it out, but you rely on it less than a more reliable cue. So this seemed to me to be quite a useful behavior and seemed to capture a lot of the features that I wanted. The pieces of information were being combined, and they were being combined in a smart way. It wasn't reflexive in the sense that as soon as the information came in, the subjects automatically made a response. And it started to seem to be a behavior that captured some of the features that would serve to begin to build a bridge from simple sensory motor reflexes to more complex behavior. However, I really wanted to study these behaviors in rodents. When I started my lab at Cold Spring Harbor about 6 years ago, I wanted to use rodent models because of the tools that are available to use in rodents that certainly at that time were not really available in other species. And I should say, at the time, people were pretty skeptical of this idea. They were open to the idea that rodents might, put auditory and visual information together, sure. But the idea that, like the humans, they do it in this very clever way, which in fact is statistically optimal, the way that the information is weighted, they said, come on, statistically optimal multisensory integration in a rodent? Like, no, it's a wild idea. But I really thought it was, but clearly I thought it wasn't a wild idea because I was willing to pursue it. And I felt that probably the machinery that existed to do that in humans was common across many species, because all species really face the same challenge of encountering an uncertain environment where some pieces of information are better than others. And all organisms have to have a strategy for determining the reliability and leveraging that in the way that sources of information are put together. So I thought about, well, how am I going to develop a paradigm that I can use in rodents? I can't very well be showing them random dot stereograms and asking them how, the height of raised bars. I need to really think about this to develop the same kind of paradigm that was used in humans, but bring it to rodents, where we have a lot of powerful tools for measuring and manipulating neural circuits. So the behavior that I came up with, I'll tell you about now. So this is a rodent in what we call a behavior box. So there's 3 ports up here, and the rodent will initiate what we call a trial. That is the presentation of some sensory stimuli about which he has to make a decision. He initiates that trial by putting his nose in a little center port there. And this triggers the onset of visual flashes, auditory clicks, or sometimes both together. And the job of the rodent is to make a judgment about whether those stimuli are high or low compared to a memorized category boundary. So we define that. It's totally abstract, and the animals just have to figure it out. They just learn with experience what means high rate and what means low rate, and they are willing to do this about 1,000 times a day, so they have lots of experience. Okay, so this is an example of a low-rate stimulus. And that one's multisensory, audition and vision together. The animal would go to the left, it would be rewarded at the left port, and here's high rate. And he would go to the right for that. So here's a movie of an animal doing the task. This is going to be a high rate auditory task, trial. And here's low rate auditory. And now you'll see a visual trial. And after this, you'll see a multisensory trial. We interleave all these different trial types together. So you can see that in those last 3 examples, they were all low-rate decisions. In every case, it was a low rate, but it was first governed by audition, and then vision, and then multisensory. So the animal is always making the same decision, but the decision is guided by different kinds of sensory inputs, or sometimes both together. And the animals are pretty motivated to do this. They really will do about 1,000 or sometimes 1,200 trials a day, and we can train lots of them in parallel. They're pretty motivated to do the task, so this affords a really good opportunity to get lots of data. And we can vary the intensity and reliability in a few ways. Just a few more examples. This is high rate. This is low rate. That one's intermediate, kind of hard to judge. And this next one's going to be intermediate and also very dim. So by controlling both the rate of the stimulus and how bright and dim they are, we can determine which cues set for the animal, which cues are more or less reliable in just the way that the experimenters did in the human paradigm. So the way that we quantify this behavior is to look at the percent correct as a function of the level of difficulty of the task. So I'll first show you, this is performance of the animal to visual stimuli alone. So when the things are very difficult, the animal is close to chance performance, but he gets up to about 83% correct. And this is the same animal on the auditory version of the task. And you can see performance is pretty well matched. Their percent correct is about equal for audition and vision. And now here's the same animal in the multisensory trial. So remember, these are collected at the same time in the same sessions, but you can see the percent correct on multisensory is much higher. And even though we have to work pretty hard to get the animals to understand the auditory and visual alone versions of the task, because it's an extremely abstract task that's quite unrelated to things they would do in their normal life in the cage, once they understand the task, this multisensory enhancement, as we call it, the degree to which multisensory is better, is not something that we have to train. They have that multisensory enhancement pretty much right from the day we present the multisensory stimuli. So then again, this really underscores the idea that putting auditory and visual information together is a very natural thing for animals to do and doesn't require training. And this is work by a former graduate student of mine, David Raposo, and we've explored this behavior extensively, both in humans and in rodents. This work is published, so I won't talk too much about it, except just to make one point about comparing the two species. This plot is going to show how well they did on multisensory versus auditory or visual. And every time there's a point below that dashed line, that means that the animal or human did better on the multisensory version of the task compared to the unisensory version of the task. And you can see that all of these points lie below the line. So it's an extremely robust effect. Every person and every rat, pretty much we've ever tested, does better with multisensory stimuli. And the circles are humans and the squares are rats. It's a little hard to see the shapes, but you don't really need to see them because they're just all mixed up together. We, in general, don't really see marked differences between the humans and the rodents at all on this task. Their performance is very, very similar. There are slight differences occasionally in the way that they use time, but overall, the degree to which humans and animals take the two streams of information and combine them is really, really similar. And this is data from a version of the task where we mismatch the auditory cues to more directly test whether the rats are combining cues optimally from a maximum likelihood point of view. And every time a point is on the line, it means that they have optimally combined the auditory and visual stimuli. And you can see that most points are pretty close to that line, suggesting that they're really close to the statistically optimal prediction. So rodents are pretty smart. They're pretty good at combining information. And when we made this realization, we thought, OK, so now we're in a good position to start to try and understand the neural circuits that support this behavior. So my lab is still really early in this endeavor. And some of the work that I won't be able to tell you about today, we're doing kind of a large-scale disruption of many different brain areas to try and understand which ones are involved in multisensory integration. But for the purposes of today, I'll just tell you about one experiment to kind of give you a flavor of the sorts of manipulations that we could do in the brain to try and understand what are the circuits that support this ability to integrate different kinds of sensory information to guide decisions. OK, so we, in these experiments, will disrupt activity by using a virus to deliver an opsin, that is a light-activated compound, to all of the neurons in the brain. So we inject it into the brain, into a part of the brain that we call the PPC, which is short for the posterior parietal cortex. And we looked at this part of the brain because we had a hypothesis that it might play a role in either visually guided behavior or perhaps multisensory integration. And back even as recently as 10 or 15 years ago, if you wanted to figure out the connectivity of a particular region in the brain, you had to kind of pore through these old anatomy papers, and they would have drawn out the sections, and they were good, but it was often very hard to follow and hard to put together. But now we make great use of the Allen Institute for Brain Sciences Connectivity Atlas, where they can inject in certain parts of the brain and tell you every place where, when you inject there, there's label expressed. And this has made it much easier for us to understand which parts of the brain connect to, for example, this area, the PPC, which is, remember, the posterior parietal cortex. So this has really changed the way that we evaluate candidate areas that might play a role in supporting behavior. So for this particular area, we had a hypothesis based on its connectivity and other features that it might play a role in decision-making, perhaps visually guided behavior. And so we first started out by recording from neurons there using electrophysiology. And we had a hypothesis that the neurons were reflecting developing decisions, maybe specifically about visual information. To get a better idea of how the neurons respond, we record from neurons. We measure their electrophysiological responses using devices called tetrodes that we build in the lab that we can put into an animal's brain and record the neurons as the animals are forming decisions. And without going into too much detail, this approach hinted to us that maybe this was the right place to look for an area that was supporting decision-making activity. But what we really wanted to do was disrupt the activity and look at the effects. So we injected a compound called chanorodopsin, which means that when it's expressed in neurons, you can shine blue light on the neurons, and they will start to fire a lot more. And this is part of a repertoire of techniques in neuroscience called optogenetics that's made it possible to, in a very specific way, alter the responses of neurons in specific parts of the brain. This is sometimes used as a technique to elevate neural activity, and in parts of the brain where all of the neurons are responding homogeneously, you can use it to more strongly drive a particular behavior. In PPC, it's quite different. The neurons there are very heterogeneous. They all do different things during these kinds of decisions. Some fire more, some fire less. Some fire in response for visual, some for auditory. And so in places like this, artificially elevating all of the neurons together will potentially overall serve to disrupt decision-making behavior. And so we wondered, well, maybe we can use this technique to test whether this area, the PPC, is really in the causal chain for decision-making. So we injected chanorodopsin into the brain, and this is to show you what that looks like posthumously after we've sacrificed the animal. Every time you see a little circle here, that means that the chanorodopsin is expressed in a neuron. And so that means that we've been successful in taking a virus and delivering the chanorodopsin to different parts of the brain. And now I'm going to show you an image. This is actually of a mouse and not a rat, but the idea is the same. You're going to see this animal making 3 decisions. And in every case, it's a low-rate stimulus, so the animal should always go to the left. And in the first 2 trials, he's going to get them right. In the 3rd trial, you're going to see a red light, which means it's an optogenetic stimulation trial. It means we're disrupting the activity on that trial, and you're going to see that the animal gets it wrong. That's low rate. He goes to the left. Good, gets a reward. Low rate again, but he goes to the right. And that's really typical. So we find that on trials where we disrupt neural activity, we see worse performance overall. So the way that we quantify this is as follows. So here on this plot, you're going to see the percent correct averaged across all of the different difficulties. And on control trials, which we call laser off trials, you can see he's at about 75% correct. And remember, some of the trials are really difficult. So we expect the overall percent correct averaged across difficulties to be quite a bit less than 100. And here you'll see what happens on the laser on trials when we disrupted the activity with chanorodopsin. And our prediction is that the animals will make less accurate decisions on laser on trials. And that's exactly what we saw. So the animal's overall performance was much worse when we disrupted activity in the PPC. So just by shining light on the animal's brain, we were able to make his performance worse. But at this point, you might be skeptical, because certainly there are lots of different ways that you could make decisions less accurate. And we wanted to argue that this was a specific effect, that this area played a specific role in decision making. And there's a few ways that we did that. And the first was to compare performance on auditory trials. So remember, all of these animals are trained to make auditory, visual, and multisensory decisions. And by comparing their performance on auditory and visual trials, we can get a sense of how specific particular effects are. And in this case, we found that performance, this is sort of the first sanity check, what happens on auditory trials. We found that when the laser was off, the performance is pretty comparable in auditory trials. Remember, they're trained extensively on both modalities. And when the laser was on, things were a little bit worse on auditory trials, but not nearly as impaired as they were on visual trials. So this suggests that either the effect is not completely specific, maybe there's some overall disruption to decision making, or the animals are just becoming confused, but, or also the alternative is that PPC plays some role in primarily visual decisions, but a little bit in auditory decisions as well. So one final way that we demonstrated that this effect was not because the animals were overall confused or disengaged from the task was to look very carefully at some of the other parameters of their behavior. And in this case, we were in a really good position because we have many tens of thousands of trials from each animal. And we looked very carefully at the distribution of how long it took them to respond and how long it took them to move. So response time is how long, once the stimulus is over, how long does it take them to pull out of the port and go off to make their choice? And then the movement is once they've left that port, how long does it take them to get to the reward port and potentially harvest their reward? So this is the distribution of response times and movement times on laser off trials. And here is, this is for an example animal, the distribution for laser on trials. And you can see that the two distributions are very much overlapping, suggesting that the animals are still engaged in the task, they're still able to report movements, but their ability, particularly on visual decisions, is altered when we disrupt activity in this particular part of the brain. So this allowed us to gain a new model for understanding how this area might work. We used to think that perhaps it had both auditory input and visual input. But once we performed these experiments and realized that we saw greater disruption on the visual version of the task, we revised that model in favor of this one, where their auditory input to the area is much weaker than the visual input. And based on other experiments that I won't have time to tell you about today, analyzing the trial-to-trial variability of neurons as the animal is making its decisions, we were also able to conclude that the integration of these events as they evolve, as they arrive over time, likely does not take place in PPC, but probably takes place elsewhere. So taken together, these experiments point to this area as one that plays a feed-forward role in processing visual inputs, possibly allowing the animal to discriminate the individual sensory events, which are then integrated at some off-site remote location, possibly in the frontal cortex, and that's something that we're trying to figure out now. So I started off by telling you that our goal was to bridge the gap between sensory motor reflexes and complex behavior, and to use that by the study of decision making. And I think that the behaviors that I told you about today won't take us all the way to the complex decisions like how to choose a car or how to pick a life partner. But in the sense that they invite animals to do behaviors that operate on a flexible time scale, and also to use behaviors that combine multiple different kinds of informations, I think they're a first step to building that bridge and can begin to take us to build on our understanding of reflexes and bridge that gap to truly complex behavior. So these are the people in my lab and the funding sources that support our work. Also lists some of our social media, including Twitter and our lab blog. And we're really grateful for all of the financial support we have to make these experiments possible. So thank you guys very much, and I'll stop there. I'd be happy to take any questions.
So feel free to get their attention.
That was a great talk. I just had a question about, you gave them two choices, so their chances of getting it right are 50-50. Mm-hmm. What are your thoughts on what might happen if the animals had three choices when you use the laser simulation? Do you think that they would then just have trouble making a decision between the other two, or what do you think might happen if there's more choices available?
Yeah, that's a really interesting question. For this particular set of experiments, we only ever had two alternatives. The work that I did when I was a postdoc working in non-human primates, we compared responses in primate posterior parietal cortex to 2, 4, and 8 choice versions of the same task. And one of the interesting things we found is that the changes in numbers of targets, it didn't change the overall conversion of sensory information into response, but seemed to change the animal's strategy or decision policy. So for example, we found that when there were more choices, that the animals tended to accumulate less evidence. That is, they kind of lowered their standards a little bit. And in part, they did that by adding a time-varying, what we thought of as an urgency signal to the neuron's response. So this is a pretty natural way to adapt to a stimulus that has more choices. If you have too many alternatives, you can't spend too long on any one, reducing the overall uncertainty. So I don't know whether that would be the same in rodents, but it would be really interesting to check.
A great talk. Son, okay. Based on the same experiment, I thought you could conclude, for example, the PPC is somewhat related to short-term memory, for instance, right? So you get this information and made a decision, but forget when it's behavior.
Yeah, I don't think I agree with the memory interpretation, unless it's a memory for vision only, because the differences between vision and audition, which we've seen also with chemogenetic disruption, I think suggests that it's playing a stronger role on the visual side. And if it was a memory related function, then that should be the same for vision and audition, because the memory requirements are the same for both modalities.
There are quite a few processes going on there. Why did you decide decision is what to look at as opposed to the learning that's going on there?
Yeah, it would be really interesting to study the learning. So one of the things that we're looking at now in the lab, so we now have a head-fixed mouse version of this setup too, that we do with 2-photon imaging. And what's nice about that setup is that we can image throughout as the animals are acquiring the task. And I think it's a really fascinating topic, particularly because this is a very abstract task. And even though once the animals are trained, we see really strong selectivity for the particular rates. We can decode the rate that was presented using different kinds of machine learning algorithms. It's very unlikely that that ability would be present when the animal was naive, but we don't know that for sure. And it's going to be, I think, fascinating to see how that population activity evolves on a long time scale of as the animal goes from being naive to expert on the task.
On one of the graphs, you had it on the x-axis difficulty of the task, and then how much it was correct. And then as difficulty increased, they answered.
Yeah, so the actual label on the abscissa should have been distance from the category boundary. So we present rates from 9 to 16 hertz, and the category boundary is 12.5. So if it's 12 and below, they should go left, and if it's 13 and above, they should go right. So because it's sort of confusing to look at psychometric functions, we often pool those two together and look at not actual rate, which is kind of an arbitrary quantity. We chose those rates based on a variety of constraints. Then it could be useful to view that as percent correct, and that's why we pool across rates that are equidistant from the category boundary. So the downside of visualizing the data in the way that I have for this talk is that it would obscure, for example, if the animal had a bias. We've looked overall really carefully to see whether animals have biases systematically on one modality or another, and also if whether that changes for multisensory enhancement, right? You might imagine, well, maybe what they do is count the total number of events, in which case they'd have a high rate bias for multisensory. But in general, and we've looked at this really systematically, they do not have bias. They do have idiosyncratic biases, but not ones that are tied to particular modality conditions. And we see animals that just as often have a low rate bias when we go to multisensory as ones that have a high rate bias.
Okay, one or two more questions.
I know it's always irritating when someone comes up and asks you about real-world applications. However, I was wondering if either you fantasize about your research being applied to the real world, so different modalities being applied to different decision making, or whether you look around sometimes and see that it's already being done. The thing that I think about is, for instance, when you turn on and back up a new car, it'll beep. And then it'll beep faster and faster and faster as you get closer to something. And that seems like exactly what you're talking about. I'm integrating both my vision from the mirror with a reinforcer that the technology that I'm using is giving to me. And it seems like that probably leads to fewer people backing into other people's cars. So do you ever, I don't know, fantasize it or realize that your research is actually happening in the real world?
Yeah, no, it's a really interesting question. I think one of the areas that I think about is related to tasks like driving, where subjects are making kind of on-the-fly decisions about some kind of action, whether they're operating a vehicle, maybe an airplane. And one of the things that we don't know very much about is kind of what happens trial to trial. And what I've showed you is that on average, subjects are much better at multisensory tasks, and that's true. But what happens on an individual trial-to-trial basis might vary quite a bit, right? Like maybe if the last, if your recent experience, in the case of the lab, maybe your last 5 trials have been reliable auditory trials, maybe when you then get a multisensory trial, even though the auditory is not very reliable, you tend to slightly overweight it because your recent experience with auditory is that it's been extremely reliable. And so it would be very beneficial in thinking about designing of cars and other kinds of vehicles if we had a deeper understanding of how our ability to integrate multisensory signals depends on recent experience. Like there might be certain states where you're very receptive to multisensory stimuli and other states where you're less receptive. And that's something that would be really fun to look at the behavioral level and also at the neural level too.
Thanks for a wonderful talk, accessible to a lot of different people. I'm interested in making better decisions. And there are a lot of ways that one can hypothesize and teaching other people to make better decisions. There are a lot of ways to disrupt circuits and disrupt decision making, and you showed some variability among different animals in the accuracy of their decisions. So as you go to more challenging environments, are there manipulations that you can do or hypothesize you might be able to do so you can help animals make better decisions or make the right decisions faster?
Yeah, I think it kind of ties into the previous question. I think I would like to understand better at the level of an individual decision, not averaged across many, but an individual decision. How is it that your current evidence interacts with your past experience? Because the framework that I talked about today was basically all about the current sensory signals, but that's not really how we make decisions. We combine those sensory signals. We don't just combine vision and audition. We combine those sensory signals with a lot of other things. For example, our prior probabilities that we think particular targets will be rewarded, or that particular streams are reliable, and so on. And so I think it's going to be hopefully possible to extend this to understand how more complex systems interact with sensory processing to guide decisions. And often, certainly in a laboratory environment, relying on those additional factors usually makes decisions worse. So this is something that humans and rodents do a lot. They tend to be biased by recent history. They tend to repeat successes and avoid failures. And this is a really bad idea, right? The trials are generated independently. So the degree to which you repeat your previous decision, it's only going to hurt you. But it's a really difficult thing to untrain out of animals and humans. This kind of superstition is a very natural thing that humans and rodents do almost equally. Just one slight difference, which is that rodents are a lot less bothered by failure. So humans have a strong tendency to switch after failures. Rodents, not so much. They're very optimistic, maybe. But I think in terms of human decision making, thinking of yourself and wanting to make better decisions, I think one of the things that's become really clear to me studying decision making, and especially in our human lab, is that people don't really know what influences their decisions. And I think we have a really strong intuition that we know what guided our decision. And we'll go back and I'll think, well, I made the decision about who to hire, based on the resume, and, it didn't work out. So obviously, it was just a mistake. But the truth is we're influenced by all kinds of factors that we are not aware of at all. So one example, we did an experiment where we wanted to distinguish rate and counting. So we had a way of dissociating those by manipulating the length of the trial. And so we could tell whether individual subjects made their decisions based on the rate of the stimuli or the number of the stimuli. We just manipulated the time over which they were presented. So we could easily tell this subject's using rate, this subject's counting, and so on. And so we asked the subjects as well at the end. And they'd been doing this task for like an hour a day for like 10 days. So they had 10 hours of experience. And we'd ask them, well, did you count, or did you use the rate? Like, what did you do? And there was no relation between what they said they did and what they actually did. So they had no idea, but often they had a lot of confidence because we think we know what strategies we're using. And I think in all of us, it's helpful to acknowledge that actually often we don't know, and we're going to try and make good decisions. We'll try and be objective and use the best information. But I think it's helpful to be realistic that all of us, I certainly can't exclude myself, that it's hard to use the best information, and it's hard to know what your strategy actually is.
All right, well, okay, one last question. One quick last question.
It'll be fast. So have you seen or heard of any differences in decision making based on rapidity of the need to make the decision or stressors involved?
Oh yeah, great question. So in general, there's something called a speed-accuracy trade-off. So in general, the faster you make your decisions, the worse you do. In terms of stress, yeah, that's something that's been studied a little bit, although less so with perceptual decisions like these and more with reward-based. And the effects of stress are kind of complicated, so I won't attempt to summarize them. But stress certainly can affect decisions, often adversely, for sure, as you might intuit.
Glad we waited for that question. So if everybody would like to join me in thanking Dr. Churchland for her talk. And we're going to segue into the second half of the evening. So as we're getting the panelists up front, I would first like to thank the members of the committee who helped to put this together, and most of them are here. So if you wouldn't mind standing, Rad Roberts, Sherry Mizumori, Bill Rooney, Kurt Haas. Did Scott make it? I know we had one other who was being taken for a Father's Day adventure and wasn't sure if when exactly he'd get here, but we've all been working together to pull together the agenda for not just the conference, but tonight. And we all felt quite strongly that we're at a moment in time in a building where we use lots of highfalutin technologies to begin to try and understand the brain, that it would be a really good time to pause for a moment and have a conversation about where we are at this moment in neuroscience time. And for those of you who aren't familiar with what we do here, by the way, I also didn't introduce myself. I'm Dr. Jane Roskams, and I'm an executive director here at the Allen Institute. So we are in this amazing time in neuroscience, right here, right now, in Seattle and across the world, where this convergence of technologies is beginning to get to a point where our field can be propelled to an entirely different level of understanding. About 20 years ago, I was at Cold Spring Harbor for a chunk of time, working with a group of people pulling together manuals and conferences that would guide the final part of the Human Genome Project. And right now feels like that time in neuroscience. The techniques are coming to the forefront. The thinkers, the modelers, the mathematicians are helping us interpret what we're doing. We are at an absolutely thrilling roller coaster ride through the brain. And now and again, we need to stop for a moment and talk about what it is that's happening and what we're doing. And I wanted to, myself and the committee wanted to pull together a few of our keynotes to come up here and give us, first of all, a few minutes about who they are, what they do, and some ideas of the way their own fields in the last 10 years have been impacted by technology advancements and developments. And then what we'd like to do is open the floor for anybody here to ask a question, even the Roberts children, who are sitting there behaving remarkably well. So I'm going to introduce our panelists for this evening. So you've already met Dr. Anne Churchland, if you would like to take your seat. Dr. Afonso Silva is joining us from the National Institute of Neurological Disorders and Stroke, and he will tell you in a couple of minutes what it is he does there. He's a senior investigator. Dr. Jeff Ogerman is here. He's a homegrown neurosurgeon who is one of those rare people who also has a highly active research program whilst saving lives on a weekly basis in surgery. And finally, it is a great pleasure to produce the person I've known longest on the panel. Dr. Linda Buck is what is your official title at the Hutch? Senior research, a member, okay, at the Hutch. Having moved there from Harvard, I met her just before she moved to Harvard when she was still at Columbia. We go way back. We both worked in the same field for quite a long time. And Linda has the auspicious honor of being the 2004 Nobel Prize winner in physiology and medicine. And now and again, we would share moments late at night at the ACHEMS meeting, coming up with strange ideas about things, some of which her people actually ended up pulling off and publishing some fantastic papers. So I think you've already heard a little bit about what Anne's doing, so I think we'll start right to left. We're going to start with Linda, a couple of minutes on what you do, and then how things that have happened, do you have your, she took things that have happened in technology over the last 10 years have enabled some of the bigger findings that have come out of your own research.
Okay, well, actually, I thought about it, and I'll say something about that, but mostly...
You're welcome to change it up.
Yeah. I thought it would be useful to give a historical perspective. And, of course, new technologies allow scientists to ask questions that they couldn't ask before. And this often leads to yet more questions and to unanticipated discoveries. Now, this is really obvious if we look back over the last century or so in the field of neuroscience. For example, in the late 19th century, Golgi developed histological stains, cell stains, that allowed Cajal to, for the first time, visualize neurons and processes within the brain and begin to understand how neurons communicate with each other to achieve brain functions. Later on, the development of electrophysiological techniques for recording electrical activity in neurons allowed the description for the first time of electrical signal generation in neurons and the propagation of those electrical signals down axons. And of course, this is one of the key features of communication among neurons. These techniques also later allowed for the identification of neurotransmitter and neuromodulator functions. These, of course, are the neuromodulators and neurotransmitters, the chemical messengers that are released from one neuron at its connection or synapse with another neuron and allow it to either suppress or excite its partner neuron. Now, these were all in the first half of the 20th century, but the last half of the 20th century also saw major technological advances that impacted neuroscience. One was in the 1970s and 1980s, breakthroughs in gene cloning and gene sequencing that allowed the identification of genes encoding many different proteins, including receptors for neurotransmitters and neuromodulators, offering the ability to use those receptors as drug targets to develop therapeutics for neurological disease, and also allowing, providing probes, genetic probes for looking at where these receptors are expressed within the brain, what neurons have these particular receptors within the brain. With the subsequent advance in sequencing technologies that led to high throughput sequencing, we saw at the turn of the century, we saw the tremendous advance of the sequencing of the genomes of human, mice, and many other organisms. This allowed the identification of genes involved in multiple different biological functions, including brain function. And importantly, it also opened the way to searching or discovering genes involved in devastating diseases of the brain, including neuropsychiatric diseases and neurodegenerative diseases. The last half of the last century also saw advances in making genetically modified animals, such as transgenic mice. So this allowed scientists to modify genes within particular neurons, affect the function of those neurons, and determine what the functions of the neurons were and what the functions of the genes expressed in those neurons were. Now, just in the last 16 years, the beginning of this century, we've also seen new technologies that are important in neuroscience. One is the pioneering development of optogenetic and chemogenetic methods that allow one to, and we heard this from Ann Churchland and her channel rhodopsin studies, that allow you to express a particular protein, a light sensing protein or a chemical sensing protein, in particular sets of neurons in the brain, and then use light or chemicals to either silence those neurons or activate them, thereby helping to figure out what the functions of those neurons are in the brain and in behavior. And then another technique, of course, and you also heard a little bit about this, is the imaging, brain imaging, which was developed in the last century but has been improved, where you can see what areas of the brain are being activated in humans. And in animals, the development of in vivo imaging techniques that allow for the observation of activity. And you also saw this in Ann's beautiful talk, the observation of activity in individual neurons in the brain during the performance of a particular task or perception. So, and as for many other scientists, I think that many of these technologies have been key to work that we've done in my own lab. And indeed, most of the experiments we couldn't have done without them. altogether as a whole. But that said, I think there is reason for optimism, as Jane expressed, but the fact is the brain is still largely a mystery. That is exactly how it functions. We've learned a lot, but there's still a long ways to go. And one can imagine a time in the future, I don't know how long it might take, but a time when it would be possible to actually visualize in some manner all the neurons in the brain and all their connections, and furthermore, to analyze all those data to really begin to understand how the brain functions as a whole to generate different functions of the mind and behavior. And of course, also to understand at an informative level what happens in diseases such as schizophrenia. So now I think to accomplish that goal, there is a broad consensus that we're going to need additional new technologies and resources. And I'd like to mention here that the Allen Institute for Brain Science at its founding, I think was over 10 years ago, about 10 years ago, recognized this need. And it decided to undertake a daunting task at that time, which it accomplished. And that was the creation of the Allen Brain Atlas, which is an online resource that shows the expression, the cellular expression patterns of over 10,000 different genes in the mouse brain. And this resource has been important to neuroscientists around the world in helping to advance the understanding of how the brain functions. And as you already heard, there's a connectomics effort here. There are other efforts occurring at the Allen Institute, and I hope we're going to learn more about those over the next few days. It's a very, very exciting place.
So I get from the middle of your answer there that if you had a dream tool, a dream thing that you could do, it would be real-time monitoring at the cellular level of brain circuits during life-awake function.
And the synaptic.
All
I
brain.
think that to fully understand, you'll need all of that, and it's impossible now. Actually, even to even visualize individual neurons throughout the brain,
right,
is virtually impossible. You can look maybe in the zebrafish, but it's very difficult. And in terms of, we study the sense of smell. So, and of course, we'd like to know, why is it that this molecule smells like orange and this one like chrysanthemums? Oh, they don't smell. Roses, okay? But how would you do that? If those are operations that are thought to occur at higher brain levels, higher cortical levels, and currently, in order to actually know how that works, I think that it's going to take new technologies to image larger numbers of neurons, and furthermore, algorithms to figure out what those neurons are doing.
Got it. Dr. Ojerman.
Well, I'm not sure where to begin, but I'll start with 25 years ago, I finished medical school. And I think it's easy to be frustrated by the pace of change in applied neuroscience or medicine. Because you look, I have the really great opportunity to see what people like yourselves are doing, and you see how far ahead it is, in just in the pure technological sense, than what we're able to deliver to patients. And so, and at the same time, many of the interventions we have are very, very similar to what existed 50, even 100 years ago, in terms of just the simple minute-to-minute tools. But when you take that longer time frame, you see a different pattern, and I think imaging is a great example. So when I started, CAT scans were very, very common. Every hospital had one. a lot of debate about, isn't it a little bit excessive to have 3 in a hospital? That seemed like too much. And not everybody needs an MRI scan, maybe, 1 in every state or something like that would be reasonable. And the CAT scan would take about 30 minutes, and that's kind of how it's used. And now, of course, CAT scans take 30 seconds to get. There's multiple MRIs in every hospital, and these things make a gigantic difference in ability to deliver care effectively and quickly, even if your individual minute-to-minute tool isn't that much different. And so when you look at many of the things that we want to do with the information that's coming out in terms of, especially as I think we'll talk about in the next couple of days, ways to treat diseases that are more restorative, so the whole neuroprosthetic push. And you look at technologies that are in your phone or in your car when you're backing up and say, well, wait a minute, I don't have that. The tools I have look like a Commodore 64, which hopefully nobody knows what that is in that section. But,
The car.
yeah, the car they'll know, but so what is it going to look like, and how do we get there, before the next 25 years? I think are the key questions, and maybe not trying to solve everything at once is also a key piece of it, because in the medical world, you're looking for victories, and you're looking for kind of small ones, because then everything else tends to come along with them. And then there's this other question, well, once you have the technology, exactly what do you want to do with it? And so one of the things that's happened with the imaging revolution is the ability to image function. And so functional MRI was really first described in about 1992, and you could, and because it was using a pre-existing technology, you could use that very quickly in people, and you could generate these beautiful images of posterior parietal cortex doing both decision-making and working memory and a variety of other things. You could look at motor cortex, you could get activities during language. But it's really, even to this day, we're putting together a consensus paper that I'm involved with. There's a lot of debate about what is this useful for, and when should you get a functional MRI, and what should you do with that information. And so that, we talk about learning a lot, and how the medical community learns is a really, really slow process. And so how that interfaces with the new technologies, with what a company might or might not be interested in, with what's cost effective, with what people want, with what people are most concerned about, with what diseases are most in need of new therapies, becomes a very complicated story. But I think it really is a unique time, because when you look at what we can do, and what's kind of in this universe of the possible, of what's only a few steps away from our existing technologies and understanding of diseases, and look at what's coming down the pike. I don't think we're going to be using optogenetics in the next 10 years. I could be wrong in people. But we are going to use a lot of the other technologies and all of our understanding that maybe optogenetics gives us about brain networks, because it isn't that big a deal to change your target of pre-existing neurostimulators. It isn't that big a deal to convert some of the neurostimulators into wireless things or turn them into smarter closed-loop devices. It isn't trivial, but that's not something that will take 10 years, I don't think. So what really should be taken out of the realm of science fiction into the near possible, I think is going to explode. And if all we have to do is get to kind of smartphone technology, imagine what you could do with different diseases with that technology.
Right.
So yeah,
that's a good
those are my
start. I'm
thoughts.
sure we'll
Yeah.
be back to you. So Afonso, if you wouldn't mind saying a couple of things about what you do research-wise, and then fire away on your dream technologies
Okay,
you'd love to have around.
Thank you for the opportunity to be here. I've actually considered myself to be very fortunate as a neuroscientist in trying to understand what the neurons are doing, and ultimately, have something somewhat intelligent about how the brain works, is to be involved in the development of many of these technologies. And for me, the technology has always been magnetic resonance imaging as the main tool that I use. It's a technology that was developed in the early 70s, and since then has seen tremendous progress in all the domains. So I like to think about MRI technology as applied to the brain as a tool that allows us to basically image the anatomy of the brain, and we're doing that really, really well. And because of the tremendous versatility of MRI in how you obtain contrast, you have contrast between different tissues that have different properties, and you relate, decomposition of the tissue, and basically, her talk was so amazing because she was able to basically give you guys a glimpse of what's important and what's being done in so many domains. But you saw in terms of anatomy that the brain has different tissues, gray matter, white matter, and within each of these tissues there is a tremendous variability in cell density, for example, and we're starting to see that more and more with MRI because of the technology developments that are being done that are basically giving tremendous results in terms of improvement of spatial resolution, to the point that MRI is almost histological these days. We can see, voxels that are smaller, even in the human brain, less than 1 millimeter cubic, and in the animal brain, less than 100 microns
Wow.
in terms of resolution. And on the functional side, again, Anne said that in fMRI, you use cerebral blood flow as a proxy for neuroactivity. We're just understanding more and more about the communication between neurons and blood vessels that allows us to understand how cerebral blood flow can be used as a proxy for neuroactivity and when it should not be used for a proxy for neuroactivity. For example, when you have an underlying neurological disorder that completely changes the way that neurons talk to blood vessels. The dream would be to, as Anne said, be able to see what each and every neuron is doing in a completely non-invasive manner, because every time you probe the brain, you are interfering with the activity of the brain. And one of the nice things about MRI is that is as non-invasive as possible. It would be great to be able to make the brain transparent, so that we could shine light into it and then see that light reflected back, into a microscope so that we could pick up the activity of each and every neuron. It's starting to be done at the level of the zebrafish. It would be great to see that done in mammalians as well. So there's a lot of hope.
Right. And Churchland, you've had a lot of time since your talk to sit there and think, what else can I add to this discussion?
I have. Well, these guys have covered a lot of territory, so I'll add maybe one kind of a different kind of technology that I think has developed, and that's in the kinds of tools that we use to analyze the data. And one of the points I argued in my talk is that it isn't enough just to collect the data. We need the right analysis tools and the right theoretical framework to think about the data. And I think we have a long ways to go in that domain, but I think there's also been a huge amount of progress in the way that people think about neural data and how to understand it. Back when I was first making electrophysiological measurements, we'd take measurements from lots and lots of cells. So we'd have their responses over time, and we'd have some much smaller number of neurons that we have now, maybe 100. And then we would take them and we would average them together. And that's one way of reducing the dimensionality, right? You go from 1,000 by 100 to 1,000 by 1, right? Because you averaged all of the neurons together. You have just one average response. But that's not really the best way of reducing the dimensionality. There's way, way better ways and lots of different ways. And it's not clear what the right way is. But now there are lots of tools that you can use to visualize your data and to think about how to make sense of really large data sets. And similarly, there are a lot of tools that people are starting to borrow from machine learning to understand how to interpret neural activity, to figure out whether it might support decisions in favor of one thing or another, or this behavior or another one, classifiers that can separate neural activity in really interesting ways. I think these are really exciting areas. One kind of a technological gap that they've revealed is that often neurons respond in really heterogeneous ways. And the way that I talked about in my talk today of disrupting neural activity it's a pretty big hammer, right? Like we artificially elevated the responses of all the neurons together. And there are currently some more precise ways to do that. But I think what a lot of these analysis tools are revealing is that populations of neurons respond in really complex ways. And one of the technological developments I hope to see that's starting to come on board now, but is in an early stage, is the ability to manipulate neurons with the complexity that we observe in their natural responses. So population of neurons responds in a very complex way. Some respond more, some respond less. They evolve over time with different temporal dynamics, some fast, some slow. And we need to be able to manipulate those responses to understand what they really mean for behavior. So that's a kind of technology I hope will continue to develop in the coming years.
Great. So before I open it up to the floor, I'm feeling the need to add a little clarification out there. Because I know everybody sitting in front of us hasn't always either heard or thought of these terms. So machine learning.
Yes. So just for example, an example of that is you have an animal making a decision, like in my talk, to go left or to go right, and you have a whole bunch of neurons that are responding. And so one example of a kind of machine learning algorithm is you take all of those neural responses together and figure out what distinguishes trials in which an animal went to the left from trials in which an animal went to the right. And maybe it's something like, well, neuron 1 fired more, and neuron 2 fired less, and neuron 3 fired in between. Or maybe it's something simple, like when the animal goes left, they all fire more, and when the animal goes right, they all fire left. But a classifier, which is one kind of machine learning algorithm, can figure out how you need to weight the neurons in order to be able to distinguish those two things. So at the end of the day, the simple ones are just ones that will figure out how you're going to combine the neurons to classify something. So averaging would just be you give them all a weight of 1. That would be the simplest kind of weighting scheme you could imagine. But there are much more complex ones, too, where neurons have different weights as well.
So it sounds like one thing we need that isn't technology is a whole bunch of creative, open-minded, math and computation-driven young people who know how to code a little bit.
Definitely, and also it's really important to have access to data sets so that people can explore. And if data are made freely available, which many people are starting to do, then people, even students in a course, can take the data and try all kinds of different things and come up with really interesting ways of analyzing the data that we haven't thought of. And if we're investing resources in collecting data, then our investments can go so much further if we make that data freely available for people to try out different kinds of ideas.
Agreed. Wholeheartedly.
And the Allen Institute has really been amazing at making data available. I think it's really been a leader in that area, and other groups are following suit as well.
They are. It's actually really wonderful to see for those of you who aren't that embedded in the neuroscience field yet. There really is a growing movement of people coming up with open data sharing platforms so we can begin to understand each other's data as well as we understand our own. So some of you must have questions for our panel. I'm all right, already at the back. And I want you to wave towards me so I can send the microphones in your direction.
So in thinking what you're saying about, we have technologies that are currently our limitation, but as the technologies become developed and we're able to then use those technologies to help analyze the raw data that we have, do you think that it's important for us as scientists in moving forward to keep all of the raw data and make it accessible to everyone so that those kind of computations could be carried out as the technology advances versus what we do now, where we, like you're saying, kind of sum up an average of everything and give it out, spit it out as a pie chart? I'm not sure if keeping all of that data available is something that we're able to really do, because it would be such a large amount of data collected from all the scientists and things like that. But it seems like there's potentially key things that we're missing because we're trying to bring it down to a very small digestible level of data.
Now, can you tell us who you are and where you're from?
Oh, okay, yeah. I'm Kenton Woodard, and I'm from the University of North Carolina at Chapel Hill.
Thank you.
Yeah, no, I think it's a great question. I think I agree with you. A pie chart is not enough information. A TIFF stack is probably too much information. I think it's a deep question of kind of what level of processing do you want to have. Like certainly at the level of one's own, in one's own data stream, you keep multiple levels and hopefully all of them as long as you can until you run out of data. But I think talking about either electrophysiology or imaging data, then presumably what people want is time varying traces for lots and lots of neurons. I think as a field we'll need to think about kind of what sorts of information would be most useful, but time varying traces, either averaged across trials or not. And I think coming up with standardized formats, it should be possible to make those available in not too enormous of a format, because having the actual raw images is probably not necessary, especially as a field if we start to get better about standardizing procedures for doing things like the early stages of image processing, like motion correction and so on. Then it won't be necessary to have the really, really raw data, and so the things that are shared will be smaller.
I think.
Yeah, go on.
I think the data sharing is a very important aspect of us moving the field forward. Tomorrow at the NIH, the Connectome research panel is meeting to summarize the main findings of phase 1 that has concluded on the human Connectome project. And I believe that two major aspects of the great success of this project were the fact that all the data that was collected anywhere in the world had to be, shared amongst all the interested researchers willing to look at that data. And second is to also establish this standard way of acquiring the data and also some common basic data processing routines and algorithms that really made the observations that, for example, I would make in looking at the data be communicable to the way that Anne would look at the same data, and I think that this is a very, very important aspect of research moving forward, is the community coming together and saying, okay, we're going to collect this data, everybody's going to be able to look at the data as they wish, and that will create opportunities for different people to see different aspects of the data that might not be entirely visible to certain individuals. And the development of these common processing tools that will make also the interpretation of the data analysis much better.
I guess I would just throw in the world of human physiology recordings, you see a range of behaviors by the investigators. And so as much as journals and funding agencies, like any good parenting, you encourage, but you also enforce. And I think there has to be maybe a little more of both the social hammer and also the money hammer of, hey, you didn't actually play in the sandbox, so you go into timeout.
Yeah. And I will say on that front, I've recently been in the middle of a number of these conversations, and both the funding agencies and a number of the major journals are getting to the sledgehammer point. And many of the reviewers, editorial board, staff really want this to happen, but the platform on which that can happen hasn't been built yet. It has in some smaller cases in a distributed fashion, but there are big movements afoot to make that happen right now in a fairly major scale way. So hopefully that larger open data sharing platform will be coming down the pipeline pretty soon.
And there are models too.
Yeah.
With genomic information.
Absolutely.
My name is Amanda, and I'm a student of neuroscience at the University of Washington.
Yay.
I feel very privileged to be in the company of the panel tonight, I must say. I desire to work as a researcher to better understand and treat neurodegenerative disorders. And as I heard Dr. Buck and Dr. Silva talk about where they felt that we would be by the year 2026 in some of their comments, I immediately thought of Stanford and the research they just published a few years ago about the hydrogel process called Clarity. And I believe that you, Dr. Silva, spoke something about that. I do know that it was done on a post-mortem mouse, but I wonder, where are we? Do we see ourselves in 10 years being able to do some sort of process like this, clarity, on an active living brain, and be able to do the things that, that Anne was talking about really being able to see how these neurons do connect within the body, and how that does relate to our behavior and making decisions. Also relating to connectome that you mentioned, again, I am a student, so one of our required readings is Connectome by Dr. Sebastian Sung. And one of the things that he does talk about in that book is the ability for all of the data that is gathered to come together for the success of the outcome. And so that was kind of my question. Do you really feel that we will be there in 10 years to where we can do something like clarity, to maybe answer some more of our questions and maybe able to map the connectome in the human brain?
Okay, who wants to field that? Alfonso?
Okay, well, I'll start a little bit. I believe that clarity has evolved a lot as a technique, and like any other technology that is developed, in the beginning you show a proof of concept, and then you work on it to make it better and better and more applicable. So one of the main points of clarity is that is a tool that is particularly useful for you to identify narrow circuits, and in a way that you try to isolate only the neurons that are part of that circuit, and you basically make the rest of the brain invisible so that you can focus and study only that particular circuit. And that's how clarity is really useful. It's reasonable to assume that the circuit doesn't change that much, even in cases of the disease, or if there is a disease, it will be useful to compare a normal brain with the diseased brain. So clarity is still used, in a post-mortem fashion for technological reasons, but it doesn't mean that it isn't useful. It's actually very, very useful, and I believe that, approaches like that should be encouraged, because I think they are making a difference. On the wish list, so Anne showed the view of the zebrafish brain and every neuron being imaged in there. That's really impossible to do in a mammalian brain right now, even in a mouse, because of all the light scattering that occurs in the mouse brain that prevents light from reaching each and every neuron and being captured back. So it would be great to continue work on clarity for that. And the connectomic work is exactly, we want to understand how the neurons are connected together so that we know that they are part of that same functional circuit, function and anatomy being very obviously linked in there. And it's a very useful approach for you to understand this neural nets and hubs, and what are the main roads and and the side streets, and how the information flows in the brain. So that's what it's all about.
I think I can also add to that a little bit. I'm on the advisory for the Brain Initiative, and in the first couple of rounds there was funding, there was a huge focus on new ways to image the living, working brain. And they particularly focused on bringing in chemists, physicists, engineers, people who'd never worked in neuroscience, but could make molecules and chemicals that would bind to certain kinds of active molecules in different ways, and the engineers to work with the imaging people on bringing a totally different perspective on how large-scale imaging could work. And already, after just a couple of years, I've seen some of the early work coming out of that, and I think in 10 years, we won't need a chemical technique like clarity, because much of this technology is going to be able to light up in different colors, different neurotransmitter pathways in the brain as it's working, that we can take to very high resolution and make it look like clarity, but inside the brain safely of somebody who may be functioning normally or not, and allow us to see what not looks like in a highly molecularly driven way. So I think that's coming. I hope it's coming. From some of the early work I've seen, it's very exciting.
Yeah, I wanted to follow up a question with Dr. Silva. I heard you mention that the MRI technology has become much more accurate and high resolution for determining neural activity, but I also heard you qualified by saying, and different mental illness disorders change the way those neural communications are occurring. Could you describe in a little more detail what kinds of changes in neural communications are happening with a mental disorder, perhaps like schizophrenia?
Oh, that's a very complex question to answer. Everything changes pretty much. You know, you have changes in gene expression inside the neurons that change which proteins get produced by the neuron, that changes the way that the neuron will interact with other neurons, change the way that the neurons will interact with the blood vessels. What is very nice is that we are starting to understand more and more of these changes, and we are developing tools that will allow us to identify when these changes are occurring. And with this, we are understanding more and more of the diseases. But it's pretty much just as much as I can say, you know, on a very complex question as yours.
But we've had some simple examples that may speak to that and its complexity. You know, when we start to use functional MRI clinically, you'll have people with brain tumors that might be very close to an area that will change with the simple motor function, like moving your hand. And sometimes when the brain tumor is very close, that activity will go down a lot, and other times it will actually be magnified. And so why one versus the other? And I think these are, I don't know the answer. But I think that even in that very simple case where you know exactly what the problem is and exactly what you're supposed to see, you don't know how it's going to interact.
Right.
So it's a steep hill.
I have a question for Dr. Silva, because you mentioned that MRI sometimes you're not allowed to say that it's actually brain activity. We use it as a proxy, right? But then you said that there was a catch, that sometimes we may not be able to actually say that it's brain activity. And I was wondering if you could elaborate on that, since there are tons of papers out there that make it seem like they just assume that it is a true proxy of it, but you said that there was a catch.
Right, so we understand really well how the neurons communicate with blood vessels in normal conditions. So in a normal brain, we understand that for example, if you do a sensory task, the area of the brain will activate, and activate being as reported by a change in cerebral blood flow. We understand the magnitude of the blood flow, the spatial localization of the blood flow. We understand all the temporal dynamics of the blood flow response. All of that basically changes with an underlying disease. In stroke being the most obvious one, you have blockage of blood flow, you have death of neurons, and the way that blood flow gets there with neuroactivity by the neurons that didn't die, is already completely impaired. So it's you can't use what you learn from the normal brain and apply it exactly to an underlying disease, because the shape of the blood flow change is different and the location is different. So that's what I meant.
Can you say anything about how similar different individuals are? Would you expect, do you actually see the same blood flow, the same readout in different individuals with the same task, for example? Or do you see variability?
Yeah, obviously there is individual variabilities, but actually when you take a fairly large ensemble of subjects, it's quite reproducible. We can identify all the sensory areas really well. If you do a visual task, you know exactly where the activation will happen. If you do an auditory task, you know where it will happen, somatosensory, etc. The overall areas of activation are very well behaved and well preserved. Obviously, the way that we study the subjects needs to be standardized so that we reduce the source of variability. It will always be there, but it's fairly well behaved.
So I would paint a slightly different picture, because I think that at least when I was doing this sort of work in my residency training in St. Louis, most of the people we studied were between 20 and 25 and wanted to get $25 for being in the scanner, which back then was a lot of money, of course. So it's, and I think that when you go into the patient population, you don't always have the same picture. And I think both the variability in the general population may be more than what we see in that subset, and then also obviously the interaction with disease that Dr. Silva alludes to. So I think you do see, and it probably, the thought, although I don't think it's settled, is that that variability is more as you get into more complicated functions. And I think a big question is how much of that is the way the brain's put together is different in these people, or are they doing the same task in a different way? And that's also hard to sort out.
Ask another question. So when you look at people with neuropsychiatric disease, do you see any major differences from normals in the fMRI?
In the fMRI, yeah.
Yeah.
Mm-hmm. And you use that information to even diagnose the disease. Yeah. I'm more in the neurological disorder side than the neuropsychiatric side, so I look a lot at stroke and I look at the multiple sclerosis. And those are...
of course, those would be...
yeah, diseases that... but they are diseases that have a very well understood signatures, MRI signatures.
Right.
And things can be much murkier when you go into neuropsychiatric disorders. For example, in autism, you have a whole spectrum of presentation of symptoms that could, really mask what you're going to find with MRI, but even how you study those patients with MRI is not fully defined yet.
So we've got a couple more questions up here, I believe. Yeah. Okay.
So you, Dr. Raskam's talked about the moving ahead of the visualization, and Dr. Buck talked about how much you'd love to see the neurons, see them, right? And what I'm curious about is whether there's consensus in the field around what kind of technologies do need to be developed. We've heard some similarities here, and what the big challenges are to making them happen. Is it just that the science and engineering is not there to have these tools developed? Are there funding issues related to moving this farther ahead? What are the big challenges getting them done?
I think people are trying to develop them, and you don't really necessarily know about it unless you know someone who's doing it or unless it's published. But just like Clarity, that was published, that was a breakthrough, and it was being done before that. So I think that there are people developing new technologies in a number of places, and we'll see those coming on board in the future, and many people will use them to ask different kinds of questions.
Hi, I work in surgical neurophysiology, and I'm just wondering, I don't know much about the two-photon spectroscopy or microscopy method. Could this be something that might be fairly quickly be used, say, to assess epileptogenic foci, or doing some kind of cortical mapping with motor, visual, and speech areas, do you think?
Well, it's a calcium indicator, so you would have to get the neurons to express it. So, like the way that we sometimes use transgenic animals. Obviously, that would be off the table. But like you could deliver a compound of interest with a virus, but it takes a while to express. There are other kinds of calcium imaging that are faster, that don't require viral transmission. But in all cases, you'd have to kind of inject that indicator into the brain. And in general, it would allow you to potentially monitor the activity of a lot more neurons, but it's also a lot slower than doing electrophysiology. So something like for epilepsy, I would imagine, I couldn't see a lot of immediate advantages over like ECOG, for example. But I think you guys should comment.
Well, one of the things that's interesting sitting here is that a lot of the incredibly cool stuff that's going on here is, and you talk about it at the neuron level, and that's really not a level that I think about a whole lot. And I would say that most of the interventions that even in 10 years we think about. And in fact, as we're sitting here, there's a meeting going on in Chicago among many of the physicians that look at the surgical treatment of movement disorders and epilepsy. And they're really able to describe dystonia now looking at population dynamics. And so, looking at the beta rhythm and how that interacts between the thalamus and cortex is different in that disease. And so it will come out of this, but I think even seeing all the neurons and their connections, I'm not sure that's sufficient, and it may not be necessary for a lot of the diseases that we have a chance of doing something about in the next 10 years. So if you look at my 10-year fantasy, it actually doesn't involve, I'm not sure I care that much about the neurons and exactly what they're doing.
Oh, but you should.
Yeah, of course I do.
You really should.
That's a different fantasy.
You know, and you will.
Yeah, exactly. But I would love to see us understand more what are these different markers and system-level markers of disease.
And so we are supposed to be wrapping up right now. I will also add to that, though. The spectrum of work being done both here and in different centers in the world right now is, particularly in the US, also funded by the Brain Initiative, is systematically beginning to develop a deep understanding of every neuron in the brain and its genetic signature, which genes it's using to function that make it different from every other neuron. So when we get that information all together, and that is a big data issue, you're really going to have the tools to actually target some of those epileptic foci and the neurons that are malfunctioning within them. Right now, you don't. You go in with a sledgehammer. But soon you may be able to go in with a laser-sharp tool, because you will understand the neurons that are malfunctioning and not the ones around them that are being damaged secondarily. And those tools will be ones that Jeff Ogerman might even appreciate at some point. So I'm being asked to wrap up, but people still have questions. Can we have a couple more? Uh-oh, I'm being told no. All right, in that case, the panel are going to be here if you guys want to, talk to them as we're wrapping up. I want to show one image on the screen as we're leaving, because this is one way that we're beginning to use technology in a different way to understand one neuron from another. We're working with the Center for Gaming Science at the University of Washington to not just not understand the genetic machinery used by every single neuron, but the morphology of every single neuron in the nervous system. So everywhere, using Clarity or other techniques, people are collecting three-dimensional, beautiful images of neurons in a circuit, sometimes groups of neurons functioning in a circuit. And they're collecting lots of data, and we don't have a hat to hang on them to understand quantitatively how they relate to each other. So coming soon to a laptop near you and your children will be a game for 3D reconstructing neurons that we need to have reconstructed in order to understand the full labyrinth of neurons in the brain. So everybody who's here tonight is going to get an email link when this goes live. And we're going to have a beta test for about 6 months to ramp it up. But this is one way that technology that you use, not the technology that we use, will be able to help us understand the brain better in 10 years from now. So we're hoping to generate a whole new generation of expert neuroanatomists from gamers of all ages, by the way. There's a couple of games we know of out there in the citizen science market where some of their best players, not in neuroscience, but in other genres, are well into retirement and doing this wonderfully on their iPad, these kind of things wonderfully on their iPads. So we hope that you will engage and contribute to the generation of new knowledge moving forward. I would like to thank all of our panel members, everybody who asked a question and showed up tonight and missed the basketball game. But it's been very close and very exciting. I've been getting updates that I didn't want to share with you because it was 1 point ahead on either side, so there were no major breakthroughs. It probably just finished. I'd also like to thank Dr. Churchland again. She's making me think completely differently about what my gut instinct is when I make decisions, probably too much with it and not with my head. And I hope that many of you will stick around and ask them questions over the next couple of days at the conference. Thank you for giving up this beautiful sunny evening in Seattle. And please go out there and enjoy it while it's still around. Thank you.